We have triacyl glycerols, and the structure of triacyl glycerol is it has a glycerol molecule and 3 fatty acids attached to it. Do you know that lipids are not true polymers? It's because if you check the structure of triacyl glycerol, see, triacyl glycerol has 3 fatty acids in it, but they are not connected to each other directly. There are 3 fatty acids in triacyl glycerol, but they are connected to glycerol molecule and they are not connected to each other directly. That's why they are not polymeric, because they don't associate with each other directly. Now we have two type of fatty acids which we have to learn: saturated fatty acid and unsaturated fatty acid. Unsaturated has double bonds. Saturated has no double bonds, which are not liquid at room temperature. They are saturated fatty acids. So why are saturated fatty acids highly flexible molecules? Saturated fatty acids are without double bonds. Saturated fatty acids are without double bonds. Unsaturated fatty acids can have one or more than one double bond within them. Okay. The unsaturated fatty acids, they exist in two configurations: cis and trans. Unsaturated fatty acid, one or more than one double bond. Unsaturated fatty acids can exist in two configurations: cis and trans. Cis configuration will have a double bond like this, but trans configuration will have a double bond like this. Saturated fatty acids also exist like this. Now, if you check their structure, saturated fatty acids do not have any room in their structure. Also, trans fatty acids do not have any room in their structure. Only cis fatty acids. Only cis fatty acids. Naturally, fatty acids occur in cis form. And cis form has room in its structure. Can you see? Lot of space is there in the structure. Because of this space, this is also called kink, the bent structure of unsaturated fatty acids. Bent structure of unsaturated fatty acids. Because of this structure, they remain liquid at room temperature. Triacyl glycerols are storage fat. They act as energy reserves. Why triacyl glycerols are better to store than carbohydrates or proteins? This is because triacyl glycerols are less oxidized than carbohydrates or proteins and hence yield significantly more energy per unit mass on complete oxidation. Okay. Moreover, triacyl glycerols are anhydrous. They are not stored along with water, whereas glycogen or other forms of storage molecules, they are always present along with water. That's why they occupy more space than triacyl glycerol. Okay, now these are common classes of glycerophospholipids. So we have phosphatidic acid, phosphatidylethanolamine, phosphatidylcholine. All these belong to family of glycerophospholipids. Enzymes called phospholipases which help hydrolyze glycerophospholipids. Phospholipid can be hydrolyzed with the help of the enzyme phospholipase A2 so that lysophospholipid can be formed. We can remove fatty acid molecule, one fatty acid from the phospholipid with the help of the phospholipase enzyme. Sphingolipids. Sphingolipids are also structural lipids. They are major membrane components, and just like in glycerophospholipids, we have glycerol backbone. In sphingolipids, we have sphingosine backbone. Sphingomyelin, cerebrosides, galactocerebrosides, and gangliosides. Gangliosides are complex sphingolipids and they have medical significance also. Gangliosides are receptors for certain bacterial protein toxins such as cholera toxin. They can also act as receptors. Gangliosides can also act as receptors. Fine?
Yes.
Steroids have 4-ring fuse structure. Can you give one example of steroid? So cholesterol is a major component of animal plasma membrane. Around 30 to 40% of plasma membrane lipids made up of cholesterol. And the steroid hormones. Cholesterol is the metabolic precursor of a lot of steroid hormones like glucocorticoids such as cortisol, aldosterone, mineralocorticoids, androgens, estrogens, testosterone, beta sterol. estradiol. So all these are steroid hormones which are synthesized from cholesterol. Cholesterol can also act as parent molecule for vitamin D production. These are called signaling lipids. Eicosanoids. Eicosanoids are derived from arachidonic acid. What is arachidonic acid? It's a 20 carbon fatty acid molecule. So the example of signaling lipids is prostaglandins and eicosanoids. Okay, can you see here from arachidonic acid? Now, what is arachidonic acid? It's a 20 carbon fatty acid. Okay. What are prostaglandins? They are signaling lipids. Eicosanoids at very low concentration are involved in production of pain and fever and in regulation of blood pressure. So what is the role of signaling lipids inside our body? They help regulate blood pressure. They help in blood coagulation. They help in production of fever and pain.